Hello. On behalf of my co-presenter, Dr. Andrew Roche, we'd like to thank the organisers for this opportunity to talk about biomarker assays, and more specifically, the application of a biomarker method in a bioanalytical setting versus the migration of that method into a CLIA lab for clinical development. But first, a brief introduction about ourselves. I have the privilege of overseeing the bioanalytical group here at ICON, whereas Andy oversees the scientific aspects of our CLIA lab, and more specifically, the introduction and then oversight of those methods in our CLIA labs. And so in today's talk, we'll have the following discussion items. First, we'll provide some perspectives about the use of biomarkers. Then we'll talk about the different levels of assay qualification required between a biomarker method that might be used in a bioanalytical setting versus one that is being used in clinical research. And we'll do that by the use of context of use of those biomarker methods. Certain methods may require full bioanalytical validation versus perhaps a companion diagnostic that needs to follow a different standard. And then at the end of our talk, we'll provide some case studies where we highlight some nuances of the application of a biomarker method depending on its context of use. And so let's now provide some perspectives on the use of biomarkers. And so let's discuss FDA's view on biomarkers. Now, back in 2014, Dr. Janet Woodcock gave a seminar to industry, and in that seminar, she made mention of biomarkers being extremely critical to drug development. And there were three types of biomarkers being discussed. And so the first class are disease-related. And so the presence of a particular biomarker may indicate your susceptibility to future disease. A good example is cholesterol. And so measuring high levels of cholesterol may indicate potential cardiovascular disease further on in life. And so in the early 2000s, industry, academia, and regulators got together and formed a non-profit organization called the Critical Path Institute. Now, the mission of this institute was to identify biomarkers that are early indicators of disease, and then correlate the progression of disease with the appearance of these biomarkers. And then there are the diagnostic biomarkers, whose presence can confirm a diagnosis of a particular disease. So for example, if you have very high levels of bilirubin, that could confirm a diagnosis of a genetic disorder, and that will be something we'll discuss in a case study at the end of our presentation. And then we have the prognostic biomarkers. Those are used to determine the feasibility of a particular therapeutic intervention and its ability to mitigate a disease. So for example, if you have the HER2 receptor and you've been diagnosed with breast cancer, then perhaps the therapeutic intervention with Herceptin would indicate a high degree of success. And then we have the prognostic biomarkers, so-called precision medicine. And so a good example here is if you are diagnosed with breast cancer and you have the HER2 and you are and then we have the prognostic biomarkers, which can be used for precision medicine. So for example, if you've been unfortunately diagnosed with breast cancer and the cancer itself has the HER2 receptor, then that provides the opportunity to intervene with Herceptin, given that its target is the HER2 receptor. And so there's a high probability that that therapeutic intervention will be successful. Now, it should come as no surprise that up to 45% of drug approvals are using surrogate endpoints. And what we mean by this is that you can measure a biomarker as to show the disease mitigation or disease progression, rather than actually waiting for a clinical outcome. So for example, you've got a high levels of cholesterol being the biomarker, and then you intervene with a statin. Now you could wait 20 years to see if the patient had a cardiovascular event, or you can monitor the lowering of cholesterol, thereby correlating the clinical outcome with that surrogate endpoint. And so the logical extension of this are companion diagnostics, where the drug company co-develops the in vitro test for a biomarker and then markets that with the therapeutic drug itself. And so the combination of both are used in a precision medicine approach. So the example before, where Herceptin use would require a companion diagnostic to measure the presence of HER2 receptors to thereby provide some assurance that the therapeutic intervention might be successful. So what types of common sponsor requests do we see here at ICON, being a clinical research organization supporting clinical development? And so within that proposal, we will see requests for And so within that proposal, there are requests to measure exploratory endpoints in the clinical protocol. And these may be standard panels, for example, cytokines. And so ICON is a contract research organization that supports clinical research. And so sponsors come to us with requests to support various aspects of their clinical protocol. And so contained within a proposal are requests for certain biomarker measurements, and they fall into one of three categories. There are those that support exploratory endpoints. And so these might be a standard panel to understand the therapeutic effect of a drug on a patient. Now, for example, we might be measuring cytokines. And so with ICON being a contract research organization supporting clinical research, we see sponsor requests for certain biomarkers. And so contained within a clinical protocol are various endpoints. Those might be exploratory, primary, or secondary. Now, exploratory endpoints often measure biomarkers as a standard panel, looking for a safety signal, or in some instances, looking actually for an early efficacy signal. Now, we might be measuring cytokine panels, or we might be looking at oxidative stress biomarkers for indication of potential harm. Similarly, we might be looking for pro-inflammatory biomarkers and their decrease with a particular therapeutic intervention. And then we have primary or secondary endpoints. For the most part, primary endpoints are typically those related to safety, although in a phase 3 trial, it's a combination of safety and efficacy. Now, the primary endpoint could be a common secondary endpoint is looking for target engagement. And so the disappearance of particular soluble protein, the soluble protein being the biomarker, would be indicative of a monoclonal antibody therapeutic binding that soluble protein and removing it from circulation. And then we have prognostic or even diagnostic biomarkers that are used as primary or secondary endpoints. You are so full of shit. And then we have primary or secondary endpoints. For the most part, primary endpoints are typically related to safety or patient safety. And then we have primary or secondary endpoints. And for the most part, primary endpoints are those related directly to patient safety. But sometimes they can be looking for efficacy readouts. More often than not, the secondary endpoints are things like the pharmacokinetics, in other words, the clearance of the particular drug, or we could be looking at a diagnostic biomarker for the appearance or disappearance of a particular indicator of disease. Another example is target engagement. So we have a soluble protein that is disease-related, and we can be looking at a monoclonal antibody whose job it is to bind that soluble protein and take it out of circulation. And then we have the class of biomarkers that are used to stratify a patient population for enrollment into a clinical trial. These are so-called inclusion or exclusion biomarkers. So the presence of a particular biomarker or at a certain level may be sufficient to exclude that subject from the clinical trial, or conversely, being from a precision medicine standpoint, the appearance of that biomarker may be exactly what we're looking for entry into the clinical trial. Let's now move on to regulatory considerations. And for this part of the talk, I'm going to be focused on bioanalytical. Now, the FDA back in 2018 issued a bioanalytical method validation guidance. And this was an update from a previous one that was issued in 2001. And so within the context of that guidance are topics that relate to biomarkers. The concept of fit for purpose is introduced, and it talks to the level of validation that may be required for particular analytes. And so if a biomarker is being measured and it is a primary endpoint, and the study itself is pivotal to submission and requires a regulatory decision, FDA are expecting that that biomarker be contained within the guidance is the concept of fit for purpose. Now, fit for purpose as it applies to biomarkers contained within the FDA guidance is the concept of fit for purpose. And this concept relates to the level of rigor that a bioanalytical method might be validated or sometimes called qualified. Contained within the guidance is this concept of fit for purpose. And fit for purpose for biomarker measurements relates to how that data is being used. So for example, if it is a pivotal study being submitted and you're looking for regulatory decision, then any method that is being used for decision making needs to be fully validated. So if you're measuring a biomarker and it is the primary endpoint for that clinical trial, then the method for that biomarker needs to be fully validated. Whereas on the other hand, if you have exploratory endpoints that it may be used for internal decision making, then the requirement for rigorous validation is not there. And then on the other hand, we have biomarkers that are being used for exploratory endpoints. In other words, more likely to be used for internal decision making than being submitted for a regulatory decision. And so in those cases, a biomarker method does not require full validation. Nonetheless, a sponsor may require of a bioanalytical lab a higher degree of qualification than might be necessary. So in those circumstances where the biomarker measurement is being used to support an exploratory endpoint, the requirement to follow all of the assessments spelled out in the guidance may not be necessary. Now, that's not to say that a sponsor may request of a contract research organization a high degree of confidence, even for internal decision making. And so it all does come down to fit for purpose and what is intended use of that data represents for that sponsor. On this slide, we provide a process flow that might be used to guide the level of assay rigor required for a particular biomarker method. One of the first things that needs to be determined is how is the clinical trial assay, the CTA, being used? Is the biomarker method being used to support a PK/PD correlation? Is it being used for enrolling or stratifying a patient population? Or in fact, is the biomarker measurement correlating drug safety? And so in the first box, we ask the question, is the clinical trial assay being used for a secondary or an exploratory endpoint? Well, the answer is yes. Then you would revert to using a bioanalytical approach, really being fit for purpose. You may or may not fulfill all of the assessments spelled out in the FDA guidance, but you would do what would be required to answer the specific question. And if the answer is no, you must ask the question, is the method being used to support patient response to the therapy, but not necessarily for a direct treatment decision? And so again, here's an example where you're talking about biomarkers that might be primary or secondary endpoints to the clinical protocol, may yet be submitted for FDA decision or a label claim, and in which case you would then be required to follow the FDA's guidance for bioanalytical method validation. Conversely, if the clinical trial assay is being used to support a primary endpoint, being used for patient selection, if it's being used for the decision to treat or as a safety endpoint, and might be considered as an eventual companion diagnostic, then the thought is that you need to undergo a CLIA validation. And that is something that my co-presenter, Andy, will talk to. The next question being asked then is the CTA being used to validate the method for a potential IVD submission, or it's going to be used as a companion diagnostic? And if the answer is yes, then there is a whole series of requirements under the CLSI requirement. Now the next question being asked is and so the next question in the process flow is whether or not the clinical trial assay is going to be progressed to an IVD, to an in vitro diagnostic, or as a companion diagnostic. And in those circumstances, you would follow the requirements spelled out in CLSI. And again, that is something that my co-presenter will touch on. And so now over the next few slides, we'll go into a little bit more depth with the application of biomarker methods in a bioanalytical context. And so on this slide, we're going to talk about context of use biomarker methods, those that are used for research use only, or in other words, internal decision making. Now that could be a standard panel of safety biomarkers, which are thought to be important for a particular therapeutic area. And that might include things like a pro-inflammatory cytokine. So if you're looking at arthritis or COPD, you might be interested in the presence or absence of certain cytokines. Similarly, if your drug's mechanism of action may induce oxidative stress, measurement of particular biomarkers might be quite important. And then in phase 1 studies, we might want to have an early readout of clinical efficacy, and so we could be looking at soluble targets and use those. Now in phase 1, where our primary focus is in safety and tolerability of a therapeutic, we may want to get an early readout of clinical efficacy. And so in those cases, you might measure a particular biomarker and then correlate that to the drug levels, in other words, PK/PD. And so in phase 1, where the primary concern is patient safety, you may still want to have an early readout of clinical efficacy. And so in those cases, you might measure a biomarker and then correlate that to drug levels. Now, the phase 1 study is often not powered sufficiently to make any clear decision on efficacy, but it could be sufficient to show target engagement and at least a hint of therapeutic activity. that biomarker may in fact become a secondary or even a primary endpoint in subsequent clinical trials. Now, that data is not likely to be used for regulatory decision, and yet it could still be included in a submission package. And so continuing on with that discussion, exploratory endpoints are often used to answer scientific questions. And so what sort of assay rigor might we use for that bioanalytical method for an exploratory endpoint? Again, just reiterating, an exploratory endpoint is for an internal decision making and not for regulatory decision. Some biomarkers might be measured with a commercial kit. A good example is a cytokine panel in a multiplexed format. Now, ordinarily, you'd want to assess precision. You'd look for parallelism if you're using an ELISA type kit, and in those cases you'd use incurred samples that you would dilute to see if upon dilution you are able to return a similar result. One thing you will want to look at is selectivity of the biomarker method in the presence of drug. Commercial kit vendors have no knowledge of your therapeutic drug and have developed their kits to measure the biomarker in the absence of pretty much everything. Oftentimes the calibrators and quality controls are prepared in buffer. And so if your drug is interacting with the biomarker, it's likely that the commercial kit may fail. Now, because biomarkers are not immediately analyzed, you'd want to understand the stability of your analyte in the matrix that it was being collected. Simply speaking, we were talking about benchtop stability and maybe a freeze and thaw cycle prior to analysis. Now, as previously mentioned, some commercial kits use buffers for the calibrators and QC levels. And so it is important to understand the acceptability of the results from those commercial kits in actual patient samples. And so what you might do is obtain commercial matrix and look for endogenous levels of that analyte, or as best as you can, over the dynamic range of your assay. Now, if you're fortunate and you have recombinant protein, you may spike on top of endogenous levels to obtain concentrations that span the dynamic range of your assay, and then you might assess that method over, say, 3 core runs. Activity of your assay is often best accomplished using the Parallelism Assessment. Now, what that looks like is that you may spike your analyte into buffer and get an instrument response, and then look at buffer diluted matrix over different dilution factors, and then comparing the signal to that buffer spike. Now, that for in that assessment is at a certain dilution, the instrument response reads back the same concentration. That's often called the minimum required dilution. So if you're fortunate and can get away with a twofold dilution, then the assay sensitivity would be quite reasonable. But there are some soluble proteins that may require 1 to 100 or even 1 to 1,000 dilution, in which case the sensitivity of the assay is directly impacted by the level of dilution needed. Now, how do we go about establishing acceptance criteria for our exploratory method? Now, clearly we could follow the FDA guidance, and for chromatographic assays, plus or minus 15% accuracy precision, or for a protein therapeutic, plus or minus 20%. And so if you're looking for fold changes in biomarker concentrations, then using absolute accuracy precision criteria from the guidance may not necessarily be appropriate. So for example, you might establish bias criteria based on plus or minus 2 or 3 times what you saw for assay precision during the qualification process. And so we've discussed what you might do for a biomarker method being used for an exploratory endpoint. Well, let's look at the other end of the spectrum. And so we're looking at pharmacodynamic endpoints that will be submitted for regulatory decision. And as I showed in the previous slide on the FDA bioanalytical guidance, any analyte that is going to be submitted for regulatory decision needs to be fully validated. Now, what does full validation mean for a biomarker method? And so what does that mean for a biomarker method validation? Well, first, you'd want to assess parallelism. In other words, understanding the impact of the matrix on the quantitation of your biomarker. You'd be looking at precision of the measurement. Now, ideally, you'd be using incurred samples, but sometimes those incurred samples may be fortified with recombinant protein. You'd be looking at selectivity, and as I mentioned before, selectivity meaning in the presence of drug, particularly if the drug is targeting binding of the biomarker. Assay sensitivity, standard requirement for bioanalytical methods, specificity of the method, and then understanding the stability of your analyte from the process from collection to analysis. In other words, you'd be looking at short-term stability, maybe benchtop or refrigerated storage. You'd be looking at the impact of freeze and thaw cycles on the analyte, and of course, long-term storage. The matrix that you prepare your biomarker QCs in needs to match study samples. So with commercial kits often being provided with using buffer, that would not be appropriate for a full validation. And so what that typically means is that you're going to screen endogenous matrix looking for matrix lots that have either low levels that you can then fortify, or you have disease samples that can then be covering the entire dynamic range of your method. Sometimes obtaining disease samples from a commercial vendor is quite challenging, and so it is acceptable to use recombinant spikes. Now, in those circumstances when your biomarker is present in matrix, it is acceptable to use a surrogate matrix for the calibration levels. Now, what is important is that your endogenous QC levels do read back reliably when you are using a calibration curve prepared in surrogate matrix. Now, throughout the validation process, you will be assessing endogenous QCs. And once you're through the validation process, you'll take then the mean concentrations from those endogenous QCs and then use a pool for sample analysis. The idea, of course, is that what you've determined during validation is replicated in sample analysis. Now, before I hand it off to my co-presenter, Andy, I thought I would show you how we've operationalized the differences between bioanalytical and CLIA. So within the ICON Laboratory Services umbrella, we've got distinct units that focus on bioanalytical methods, as well as the central lab doing traditional CLIA safety testing. And where there is overlap, we refer to that as specialty services. So for example, we might have a traditional pharmacokinetic assay where we're measuring the large or small molecule using ELISA or LCMS. But in that same group, we might also do biomarkers, given that the platforms are identical between doing a drug versus the biomarker. And so where we have overlap, we call that group specialty services. You might have a biomarker that might have been exploratory and done in the bioanalytical lab, is now potentially becoming an enrollment criteria, and that needs to be then done in a CLIA lab. And so we would transition the assay from the bioanalytical setting and then into CLIA, which is what Andy will talk about next. And so in upcoming slides, Andy will now discuss what is required for a biomarker method transitioning from a bioanalytical setting to now into clinical research, where the results from the biomarker measurements are going to be used for patient enrollment, inclusion-exclusion criteria, or be reported to the clinical site for dose decisions.
To discuss the factors that are important to consider when reporting results to clinical sites. Firstly, for US-based labs, it is essential, and it is a federal requirement, that those labs have CLIA accreditation. In addition, many labs, for example, including those participating in clinical trials, will also opt to obtain and maintain the College of American Pathologists, or CAP accreditation. This is an industry standard accreditation which builds on the requirements of CLIA. CAP have a mission of fostering and advocating excellence in the practice of pathology and laboratory medicine worldwide, and accreditation by CAP also covers off your CLIA requirements. A further point to consider for labs based in New York State, or for labs who wish to report results to citizens of New York State, it is also essential in those scenarios that the lab obtains either a New York State permit or a license such that they can report results for subjects based in New York State. In addition, the CLSI, the Clinical and Laboratory Standards Institute, also provide a number of guidelines that can provide more detail and more guidance on how to perform specific studies. Typically, these guidelines are of, I suppose, more specific benefit in designing the exact experiments, as compared to CAP or CLIA, who will tell you, for example, that you must do a study X or study Y without actually saying how to do that study. So CLSI are very valuable in that respect. They provide various procedures that cover methods from start, the lifespan of methods from start to end. And some of the particularly useful methods that many labs would use would include, for example, EP28, which defines the establishment and or verification of reference intervals in the clinical laboratory setting, and also EP15, user verification of precision and estimation of bias. So these documents are very helpful to use from a clinical laboratory perspective. When considering exactly how to validate or verify a method, when setting one up in your clinical lab, I suppose the first thing is to consider, do you need to verify or do you need to validate? With that in mind, CAP provide a definition for both. So analytical verification, as per CAP, is the process by which a laboratory determines that an unmodified FDA cleared or approved test performs according to the specifications set forth by the manufacturer when used as directed. Analytical validation, however, is the process used to confirm with objective evidence that a laboratory developed test or modified FDA cleared or approved test delivers reliable results for the intended application. So that's what the key distinction there is. Verification is for IVD tests that are used exactly as per the manufacturer's recommendations, or if you're not using an IVD test, then you need to consider analytical validation. When validating laboratory developed tests, I suppose there are a number of different factors to consider. Firstly, those developed tests can be either developed in-house. It can be based off analyte-specific reagents. It can be based off research use only reagents or investigational use reagents. And the extent of validation should be sufficient to ensure that you have confidence in the results that are released by the laboratory developed test. One very important factor to consider with laboratory developed tests is the risk that the result carries with it. And this is a factor that the FDA have been looking at over recent years. And some of the high risk areas that are worthy of consideration include, for example, will the test provide a direct standalone result that guides a specific therapeutic intervention? Or is this a screening device for serious diseases and or conditions intended for use in asymptomatic patients with no other available confirmatory diagnostic product or procedure? In those scenarios, it is essential that the result which is provided back to clinical site is of high accuracy and one that the laboratory has confidence in. A failure to do that may result in detrimental events for the subject in question. So when considering validation or verification, labs such as Icon Central laboratories who are 100% dedicated to global clinical trials will typically focus on ensuring that the method is capable of creating high quality analytical data that is analytically precise, analytically accurate, and complete, i.e. there are no gaps in the databases, or the presence of gaps or less than values or greater than values is minimized. The reason why at least Icon Central Labs focus on this is that by creating such high quality databases, we are able to pass back to our customers high quality database that enables them to efficiently and accurately assess the safety and efficacy of the compound of interest to them. And in order to do that, we spend quite some time considering how to validate or verify the performance of an individual assay. On screen at the moment, you can see the typical format that we will use when it comes to verifying or validating an assay. So firstly, we will typically perform at least 6 assays, and we will involve at least 2 operators. And then precision experiments, for example, will be performed typically in accordance with EP15, using typically 5 reps per level over at least 4 levels across at least 5 assays. The typical concentrations that we will focus on will include concentrations that sit at the upper level of normal and lower level of normal at 3 times the LLOQ and 0.7 times the ULOQ. And routinely, the acceptance criteria that we assign are based off a total allowable error-based system, or TEA, and we usually expect the precision of our methods to fall within 1/3 of the TEA. Then moving on, we will perform accuracy, linearity, and reportable range study. It's usually the same study performed with 5 separate concentration levels, 5 reps per level in a single assay. We will expect to see appropriate levels of linearity and accuracy within that experiment. Acceptance criteria here are usually 50% TEA, and then we will also perform accuracy assessments with, for example, CAP proficiency samples wherever available, or with international standards wherever available, or in the case of laboratory developed tests, we will try to perform comparison studies with another lab performing the same method, using 20 or more samples. The acceptance criteria here will typically be focused on those applied with CAP for the proficiency scheme, or based on 50% TEA, for example, when comparing with another lab. Carryover experiments are important to perform when using automated devices where the sampling involves a device that will go into every sample, and where what you're looking for is to ensure that there is no carryover of a high sample into a subsequent low or negative sample that inaccurately causes that low or negative sample to now report out as positive. And carryover studies are performed in accordance with, again, with EP guidelines and with EP10. Moving on then, other studies that will often be performed depending upon the features and the needs of the assay include, for example, dilution and linearity assessments, where samples will be diluted to confirm that the actual dilution step itself does not impact the accuracy of the result. We do, as a global laboratory, perform bias assessments between our respective laboratories. So before a new lab or a second lab brings up a method, we will expect them to be harmonized with the primary location before they start reporting out results. This is, I guess, not so much important for individual laboratories, but it is important for Icon Central laboratories, whose one of his goals is to provide harmonized results across all locations. And we use CLSI EP9 to guide these studies, which effectively requires that you run at least 8 samples over each of 5 days as a means of accurately assessing the bias of that exists between the 2 respective assays. As mentioned previously, reference range verification is performed in accordance with CLSI EP28. In this document, you can either verify a reference range that exists, for example, in a product insert, And to do that, you can run 20 normal samples, and if 90% of them come back with the expected result, you can verify. However, there is also an option to establish a reference range, in which case CLSI recommend using 120 samples or more to establish the range of results that would be expected to be seen in a normal healthy population. Stability studies are often required, and with an ICON we use a total change limit approach for performing these studies, where one considers both the analytical variation of the assay and the biological variability of the analyte as a means of identifying acceptable levels of drift for an analyte before, identify that the levels of drift are acceptable before a result should be cancelled out. And then, of course, selectivity and specificity studies can be performed where necessary, and where, for example, a drug being used in a study may be suspected of interfering with the result, or where a comparable, for example, biological agent, where there is a risk that may cause a false positive result as well. And they are performed in accordance, at least with an ICON, in accordance with the FDA Bioanalytical 2018 criteria. Once one finishes the validation process, it is of course essential that the focus on maintaining the quality of the assay does not stop there. With this in mind, there are many different procedures available to ensure continued control of the quality of individual assays once they go live. With an ICON, we use quite an evolved system, which includes, for example, daily QC monitoring, weekly QC monitoring, monthly QC monitoring, based off the performance of the assays when controlled with an analyte-specific alkyl level error and with Westcard rules. Parts of this process that are worthy of consideration include, for example, being sure that you can control the precision, the bias against the target. And there are, of course, numeric values that can be used to track performance of precision and bias against peers using, for example, the SDI value for assessment of bias and CVR value for assessment of precision when compared against peers. As one would expect, the ability to maintain quality of assays is highly reliant on the inputs into the system, because adding high quality inputs will maximize the ability to drive high quality outputs of that system. So with this in mind, for example, with an ICON, we would start off by trying to select the highest performing assay for a specific analyte. So, for example, it would, I'm sure we all, most on the call, have experiences of situations where automated assays will create typically higher quality assay than, for example, manual benchtop assays. So, for example, the CVs of automated assays will almost always be 1 to 2-fold tighter than the CV of, for example, assays that require manual pipetting into plates. It is also essential that QC rules are assigned from the very start, that training is completed before any individual starts to run the assay, and that devices are consistently and accurately maintained. In addition, it's important to have incoming acceptance criteria for new lots of critical reagents. And by creating or by driving high quality inputs and by monitoring them before you introduce them into the analytical system, you maximize your chances of creating high quality analytical data outputs. With an ICON, we are very fortunate to have a global QC group who will monitor the performance of assays over time, and that enables us to ensure the continued high quality performance of our laboratories. In addition, we also participate in, for example, CAP proficiency schemes, which provide observational monitoring opportunities where one can assess at a given point in time the accuracy and precision of that laboratory against other laboratories also participating in that scheme. some of the, as the purpose of the slide is to illustrate some of the features of our current assay maintenance quality procedures. On the top line, you can see the assay quality component being QC, and this is probably the most important one that we use on a day-to-day basis. Using QC enables you, one, to assess accuracy within an assay, precision within an assay, but also it enables one to compare between different devices within a single lab that are running the same assay, and also between different labs that are running the same assay within our laboratory network. And this is very helpful in the context of trying to identify any analytical systems that are at risk of going out of specification. Typical calculations that can be used to identify analytical systems that are going in that are at risk of going out of specification include, for example, the Westgard rules, which enable detection of drift, and also there are CVR and SCI calculations which can be used to assess precision against peers and bias against peers, respectively. As mentioned earlier, CAP proficiency surveys also provide a lot of benefit in that they let you compare between laboratories in your network, but also they let you compare yourself against laboratories outside of your network. And assessments against both precision and bias can be made with these samples, albeit they are predominantly used for bias. The assessments that are listed, you can see in the third row of the table. In addition, then lot-to-lot is a key component as a means of ensuring that the reagents that are brought in are consistent, are performed in a manner that is consistent with those used previously. With an ICON, we assess lot-to-lot using a 1/3 TDI criteria, so we would not expect a new lot to introduce a shift of more than 1/3 of the analyte-specific TDI. And among all of those assessments, it's important to note that the use of the TDI-based system does enable you to put a single measure of analytical performance on each and every assay, and the process signal value enables you to do this. With an ICON, we aim that all of our assays hit a sigma value of 5 or more. We would have liked them all to be meeting the 6 sigma standard, but it's not possible for all assays. And the minimum standard that we target is there should be no assay performing below a process signal value of 3. So moving on, so we've talked about IVD assays, we've talked about LDT assays, and there are another subset, the companion diagnostic assays. So what are companion diagnostics? Well, companion diagnostic devices can be in vitro diagnostic devices or an imaging tool that provides information that is essential for the safe and effective use of a corresponding therapeutic product. The use of an IVD companion diagnostic device with a specific therapeutic product is stipulated in the instructions for use in the labeling of both the diagnostic device and the corresponding therapeutic product, as well as the labeling of any generic equivalents and biosimilar equivalents of the therapeutic product. The guidance is issued by the FDA. There's quite a number of them. Some of the key ones that people interested in learning more can access are listed on screen at the moment. One of the key factors associated with, with companion diagnostic co-development with a therapeutic product is to consider the level of validation that should be performed on your IVD CDX device, and the stages at which that validation should be performed. In the early stages or in preclinical stages, having a highly developed product is not necessary, but it is essential that when you reach the end of when your product reaches the end of its pipeline, when you aim to get marketing approval for it, at that stage it is essential that you have a highly developed companion diagnostic product which will meet the FDA's requirements. And as such, it is important to consider how you develop your product and the stages at which you do that, because it's critical to ensure when you reach the end of when you reach the stage where you're going to apply for marketing approval, that you have a ready to go companion diagnostic assay that has been developed to the standard required by the FDA. So with that, I will pass you back over to Roger.
Thank you, Andy. So now let's move on to some case studies. And we've selected these case studies because they are not immediately obvious as to whether they should be performed with a bioanalytical method validation or executed in a CLIA lab setting. So in our first case study, we're looking at cytokine analyses. Now, typically cytokines are looked at in an early phase where we're trying to determine if there is a safety signal for an up or down regulation of a particular cytokine or a group of cytokines. And so therefore you would consider a bioanalytical lab to be more appropriate in doing the initial assessments and potential reporting for internal decision making. But once particular cytokines have been identified as being disease or safety indicating, then those may well progress into a CLIA lab setting. Now, the instrument platforms that you might use may be entirely identical. So for example, in ICON, we would do cytokines on a Luminex system, which is a bead-based approach, or on an MSD electrochemiluminescence system using the VPlex. And so a very common example is the cytokine panel shown on the right, being a pro-inflammatory panel for exploratory phase of clinical development. So here in our second case study, we're dealing with a genetic disorder. In this case, it's a syndrome called Crigler-Najjar, where the unfortunate individual has an enzyme deficiency, being the bilirubin UGT enzyme. And so the body clears bilirubin through glucuronidation using the UGT enzyme. And if you're unable to clear bilirubin, it will build up in the body, leading to jaundice, but in newborns, you can lead to brain damage and ultimately death. So in this example, we have two analytes. On the left-hand side, we have bilirubin itself, and then on the right-hand side, we have the glucuronidated version of bilirubin, being the glucuronide. And from a clinical standpoint, it would be ideal if we were able to measure both bilirubin and its glucuronide. Diagnostically, if you have high levels of bilirubin and no levels of bilirubin glucuronide in circulation, you might suspect an enzyme deficiency such as the Crigler-Najjar syndrome. Now, upon therapeutic intervention, in this case, you might look at gene therapy, where you are now adding or replacing a deficient enzyme with the UGT1A1 protein. You can monitor the levels of bilirubin and look to see if the glucuronide form starts to appear in circulation. Now, because we're dealing with pivotal pharmacodynamic endpoints, it seems pretty clear that we would follow the FDA bioanalytical guidance for method validation. Now, there is a nuance, because if the patients being selected for gene therapy are being screened based on their bilirubin levels and lack thereof of the bilirubin glucuronide, then there might be a thought that those methods be performed in a CLIA laboratory. Now, because it is gene therapy, you normally think of that as a one and done. And so the potential for therapeutic dose decisions based on the bilirubin glucuronide levels may not be as important. Let's now move on to case study 4, and we stick with gene therapies yet again. But here we're looking at the vector delivering the gene therapy. So the introduction of a transgene, being the messenger RNA, into a cell is often affected by the use of an adenoviral vector, or in some cases a lentiviral vector. Now, there are at least a dozen different subcategories of adeno-associated viruses, and a drug company will select a vector that is most suited for the particular target tissue that they're interested in. Now, because the vector is a virus, much like any virus in the wild, there may be individuals in the population that have been pre-exposed to that virus and thereby have antibodies to the viral vector. And so therefore, if that individual is being dosed with a transgene encapsulated in that viral vector, it is likely that the body's immune system will react and clear the vector before it's been able to transmit the transgene to the target cell. In other words, from an inclusion or an exclusion criteria in a clinical trial, you would want to pre-screen patients for those that do or do not have the viral antibodies. That being said, when you're assessing immunogenicity, that is quite commonly done in a bioanalytical laboratory. And so in this example, the immunogenicity assay may well be established in a bioanalytical lab, but when it is being used for screening criteria in a clinical setting, then it needs to migrate to a CLIA lab. And so our approach at ICON is to develop the initial screening assay in the bioanalytical laboratory following the FDA's immunogenicity guidance and, of course, industry best practice. And what is quite common for a screening assay is that you're looking for about a 5% false positive rate. Having a false positive rate would mean that a small percentage of the population would be excluded from treatment, but from a conservative aspect, you would not be treating anybody that had the potential for pre-existing antibodies to the viral vector. And so once the screening assay has been developed and then validated in the bioanalytical laboratory, we are now ready to transfer it into our CLIA lab. Now, we would consider the bioanalytical method validation sufficient for establishing a lab-developed test in our CLIA laboratory. We would then undergo the requirements of a CLIA validation, and then because ICON's central lab exists in the state of New York, we would submit to New York State Health for final approval. And so once efficacy has been demonstrated for the therapeutic intervention, the next logical step would be to collaborate with a reagent vendor to develop an in vitro diagnostic to then, in clinical medicine, be able to pre-screen those patients that are predisposed to having antibodies to the viral vector, and thereby not subjecting them to a therapeutic intervention that would have little chance of success. And so that concludes our case studies. And so hopefully with these examples, you are able to determine for yourselves when a CLIA lab setting or a bioanalytical lab setting might be more appropriate. And so with these case studies, I hope we've shown that it is not a clear-cut decision as to whether a bioanalytical lab or a CLIA lab should be performing the biomarker method development and validation. And with that, we'd like to conclude. And on behalf of my co-presenter, we hope that you found this presentation informative. And if there are any follow-up questions or feedback, please feel free to reach out to us, and our emails are presented on screen. Thank you for your attention.